Let me start with this one. So here's gonna be the first question, everybody. Ooh, sorry, sorry. I know I'm no longer screen sharing. Let me fix it. Okay. Tell me when you can see my screen. I'm trying to make you all see my screen. You can see my screen now, right? Good. So here's the first question. Right, let me just say there are things that I need to say, of course, before I start answering. That are things that we use, things that we apply, things that we don't break. So listen closely, everybody, and let's just hammer this as quick as we can. Fast. So the idea is going to be this, okay? Listen. When you are doing Punnett square questions, remember there are things that we talked about that you have to know. There are things that we talked about that you have to make sure you know. The things that you have to all of you make sure you know is that what is heterozygous? What is homozygous? What is the question asking about? Is it asking about codominance or whatever? What is it? So there are things that, and how do you write phenotypes? How do you write about genotypes and all these other things? Okay? Are you listening? So let's start with this one here. The first question says the allele for the normal form of hemoglobin is this one. This is for the normal form. Then for the abnormal form of hemoglobin is this one. So this question had already started to talk about it, a condition called anemia. Okay? Condition to do with blood. So, and it's saying these are the alleles for this. Now let's see what it says. Draw a genetic diagram to determine the probability of what type of parents? Let's find out about these parents. Probability of two heterozygous parents. What an easy thing. Two heterozygous parents. Okay. Let's do for two heterozygous parents. So if we come to the genotypes of the parents, we've been told they're heterozygous. So you say HBA, HBS, HBA, HBS. These are heterozygous, which means both parents have both alleles. These parents, if you're heterozygous, it means you have both alleles, right? So now they said, tell us the gametes. The gametes you just separate. There's HBA, there's HBS. Then there is HBA, and then there is HBS. So now let's do it. What are we going to do? They say draw the probability, so it's just a Punnett square. You now need to put your Punnett square. Remember how you do your Punnett square. You say 1, 2, 3. 3 lines like this. And then you put 1 again, 2, 3 like this. 3 lines as well. Right? Now, let's go. This is going to be HBA and HBS. Then HBA and HBS. Now cross the parents, please. This is so easy. HBA meets HBA, you get HBA, HBA. HBA meets HBS, you get HBA, HBS. HBS meets HBA, you get HBA, HBS. Okay, you always start with the dominant one. HBS meets HBS, you get HBS, HBS. Because it's this one meeting this one. And you get the results in here. Now, what are they saying? Find the probability of what? Having a child who does not have the HBS allele. Yes, you can ask me your question. Yes. Ask me your question. Somebody called my name. You can ask me the question. Is it you, Michelle? Michelle, what's the question? Or are you agreeing or what? Oh, you can't see my screen. Check on YouTube, Michi. Or can others see my screen though? Can the others see my screen first of all? Okay, Michi, check on the YouTube page. Or try to rejoin. Maybe it's the network because the weather is definitely not good. Okay, Michi. Michelle? Yes, try to rejoin. All right, I should continue. Michi, you can try to rejoin or check the YouTube page. Go to live videos, you see it. So now let's write. So for this part, is everybody fine? Students? Are we all fine on this? Now look at what they said. Somebody needs to mute the mic. Can you all mute your mics, please? Thank you. Now here it says, no, mute the mics, guys. Let me see where it is. All right. So now we'll be saying, look at this. Give me 1 second. Okay, we'll be saying here the question. This is the easiest question. So if it's just us constructing Punnett squares, this question is way too easy. We can do it. What are the offspring genotypes? They're asking us for the genotypes of the offspring. We can see that we have HBA. Okay, they're asking us what do you get as the offspring. Remember, the genotype is what you see inside here. These are the genotypes here together. So we have HBA. Do we have another one? No. All right. So we have now HBA, HBS, and another one again. So you can put it like say HBA, HBS, another one, because there are two of them. Then you now have finally HBS, HBS. This is the very last one which you have. So here we had two of the same type. So the other one, we usually put it in brackets when it's two of them like that. Now, what's the probability of not having the HBS allele? How many of these don't have the HBS allele? It's only this one which does not have an HBS allele. So the probability is 1 in 4. You can write it like that, to say 1 in 4. They allow you, or to write it as a percentage, to say 25%. If it's 1, it's 25%. Are we together? Here, if you come to where they say parental phenotypes, now they're saying, what do you see in parents? So here, if they are heterozygous, then they are carriers. So these parents are carriers of whatever this sickle cell disease. So you can say they are carriers. The carriers of this. So they're carriers of this. So you can say these are unaffected parents or they are carriers. So this is a carrier and another one is a carrier as well. This person, because for you to have it, obviously, it needs to be there here as HBA. Then you would have it. The HBS rather. Then you'd have it. Any questions on this? This is easy, right? Class? Any questions? Right, let's do the second one. This is now codominance. When you see blood groups, they've already given you things. They just want you to do the Punnett square. So we have the genotypes, IA-IO. So we can write it even. If you want to put IA-IO here, it's still fine. Then put IB-IO here, it's still fine, wherever you want. Now IB meets IA, it will be IA-IB. Now IB meets IO, it will be IB-IO. Now IO meets IA, you write it as IA, then IO. IO meets IO, you write it as IO, then IO. Now they say, what are the phenotypes of the children? The phenotypes, this one I taught you in codominance to say, when you have these two, they express together. So the phenotype will be blood group AB. Then we said when you have B and O, B is dominant when it's like that. So it will be B which is shown. So person has blood group B. If you have A and O like this person will be blood group A. So it will be like this. If you have IO, then the person has blood group O, which is in here. So these are the 4 phenotypes that you have. Are we together? Are we together? What is the probability that the child will have blood group O? You still again say 1 in 4 or 25%. Are we together, students? Can you hear me? Good. Person 7. Oh, this question. Okay, let me explain to you. Person 7 was a pedigree chart, and person 7 was shaded to say they are affected of a disease. Sorry for not sharing. So this is going to be a bonus one when I mark. It's bonus. So person 7 was shaded to mean they are affected. So now they are affected by cystic fibrosis, which means that person 7, if they are affected, what does it mean? They have both alleles A and A, because they said A is for recessive. So they'll be having both alleles A and A. That's person 7. So they have both. That's the only way they'll be affected, because A is the recessive. For it to affect you, it should be the only one there. So it's A and A. So person 7 is this one. Then who are they? A man having a child, person 7, and a heterozygous man. So person 7 was a female, which means it was affected and it was female. It was circle and it was shaded. Now, having a child with a heterozygous man, which my screen is what? Blurry. You can't see. Others, can you see my screen? Oh, so try to leave and re-enter. Now here we have a heterozygous man. So these are the alleles of a heterozygous person. They will be having both alleles. So it's A and A. Now the gametes, small a, big A, small a. These are the gametes. Now what do you do? There we go. What do we do now? We make obviously our Punnett square. This is the easiest thing. 3 lines, 3 lines. Don't forget. 1, put it like this to say 1, 2, 3. Then you start here, you say 1. Okay. Then you say 2. And you say 3 ending here. This is your Punnett square. You put your alleles A and A, then big A and small a. Now you start crossing A and A. Okay, usually we like to put the farther this side, so you say A, big A and small a, then small a and small a, still fine. Big A meets small a, it will be big A, small a. Big A meets small a, it will be big A, small a. Small a here meets this small a here, it will be small a, small a. Small a here meets small a here, it will be small a, small a. So now, what are the genotypes of the offspring? It means the things inside your Punnett square. Those are the genotypes. We have what? We have A, okay, big A, small a, and we have 2 of them, so you can put it like this. Then we have small a, and we have small a again. What are the phenotypes of the thing? The phenotypes are, if it's big A, small a, that means that is what is expressed, the big A. And what does big A represent? Is this person affected or not affected? This person in the phenotype, they don't have the cystic fibrosis if they are heterozygous like that. So what are the phenotypes? The phenotypes are without cystic fibrosis, because we are dealing with cystic fibrosis, without cystic fibrosis, and small a, this one has, so it's with cystic fibrosis. So we have people without cystic fibrosis, then we have people with cystic fibrosis. Probability of having a child with cystic fibrosis. How many children have cystic fibrosis? Those with the recessive alleles, only 2. So you can say it is 2 in 4, or 50%. That's the probability. Are we together? Are we together, students? Okay, good. Next question. Question number 4. Can you see? We're on question number 4 now. We are continuing. We're on question number 4. What does it say? Question number 4. It says, select a suitable letter, blah, 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 blah. We have 71 offsprings and 26 offspring in yellow. What is this ratio? We have ratios. Do you remember the important ratios that we talked about last time? Right. Let's put. That is one of the most important tools that you can use when you're answering in here, in case they are testing you. We have the ratio 3:1. Then we have the ratio 1:1. Do you remember? And here we are talking about here the cross produced with red fruit. This is what we call phenotypic ratio, because they are talking about what they looked like. They had red fruit. These ones had yellow fruit. So we have 71 to 26. 71 had red, 26 had yellow. This is almost 1:3, isn't it? This is almost like a 1, is approximately like a 3:1 ratio. Is it not? What's 26 times 3? It's in the 70s, right? So this is like a 3:1 ratio. Are we together, students? This is like a 3:1 ratio. Now they're saying, what is letter representing the allele? Here they select, okay, select a suitable letter to represent the alleles. Which allele we need a capital letter? We can use letter A. So what letter will represent the allele for red fruit? We'll say letter big A. For yellow fruit, we'll say small a. Now they're saying, what are the genotypes? If something is red fruit, and then here this one also had red fruit, so obviously these are heterozygous, because they should give a child with yellow fruit. We know that when you cross heterozygous, you should get the phenotypic ratio 3:1. So let's prove that. Let's prove that. Let's prove that. I wanted people to know. So do you remember? I will explain here again. When you cross two parents with different alleles, the phenotypic ratio should be 3:1. Do you have the notes where we wrote that? Students? The gametes here will become a big A, small a, and big A, small a. Now we do our Punnett square. We do 3 lines like this. 1, 2, 3. Then we do 3 lines like this. We put 1, 2, and 3. Now we'll say big A, small a crosses with big A, small a. We cross big A, it will be big A, big A. Then this big A meets this small a, it will be big A, small a. Then this big A meets this small a, it will be big A, small a. Then this small a meets this small a, it will be small a, small a. That is why we say the ratio is 3:1. 3 red fruits, you can see them. This one is obviously red because the dominant is there. This one is red, the dominant is there. This one is red, the dominant is there. This one is yellow because there is no dominant. So we'll now say, they're saying offspring genotypes, we have AA, then we have A, big A, small a, which again we have 2 of them. Then we have small a, small a. Now what is the phenotypic ratio? 3 red fruit as to 1 yellow fruit. That's the phenotypic ratio. 3:1. That's the expected, which is almost the same. So this is what you do when you have heterozygous. How did you know we have heterozygous? We have heterozygous. They said, they talked about the ratios. You get 71, 26. That's a 3:1. So you know it's heterozygous there. Are we together? Can you imagine that just doing this gives you all these marks? 6 marks. Just doing this. Can you imagine? The gene for red color vision is on the X chromosome. So can you see that they're talking about sex-linked characteristics? Sex-linked characteristics is when you have a gene on a certain chromosome. Right, guys? So now, there are 2 alleles for this gene. There are 2 alleles for this gene. The allele for normal color vision is big B. For less, it's small. If it is on the X, hello? Then we would say X with big B and X with small b, because it's on the X chromosome. Once you talk about sex-linked, these letters, you are putting them on the chromosome where they are found. Are we together? So here now, what do they want? So they've told. Let's go to the question. Parent 4 is a female with normal color vision. If they have normal, remember that red-green color blindness is small b. Normal color is B. So parent is a female with norm and is heterozygous for red-green. So if it's heterozygous, female with normal color vision, but they said she is heterozygous. So you say, remember, it will be X big B and X small b. Females are XX. Now, a male that is colorblind will have the colorblind X, which is small b, and men, we know that they are XY. But because this man is colorblind, the B he has is not the big B. This one is for no color blindness. He only has the one for red-green color blindness. So he is colorblind because he's going to be like this. What saved this female is the fact that she had the dominant one as well. The man has a Y, so he doesn't have the dominant one to pick this small one. Now let's put the gametes X, big X and small b, and then a Y. Then let's put these ones, big X, X big B, and X and small b. Now let's cross them. So we're going to say 1, 2, 3. Then we say 1 like this. Then we say 2. Then we say 3. Now let's put the things. We know that for males we put X and Y. For females we put X and big B and big and the X and the small b like this. So showing this knows that you understand sex-linked characteristics. Are we together, students? Now what do you say? X with big B crosses with this, you get X big B, X small b. This female is a carrier. She doesn't have color blindness. X small b mixed with X small b, it will be like this. Now, this female is colorblind. It's a red-green colorblind, which is very rare. Big X means this, you get like this. This male is not colorblind because he's going to be XB with Y. But this Y will meet with this X, and you are going to get a boy that is colorblind. He is XY. Women are XX, boys are X, males are, females are XX, males are XY. So here, he is colorblind because the X he has is for color blindness. Now, what is the thing saying? Offspring genotypes. Let's put all the genotypes. We have X big B and X small b, which is that first one. Then we have XB small b and X small b. Now, we now say another one, we have X big B with Y. Another one, we have X small b with Y. These are the genotypes, the solutions that are in the solutions that are in there. So now, what are the phenotypes? What are the phenotypes now? Let's put the phenotypes. What do you get here? Female carrier. Are we together, students? Carrier is a person who is heterozygous. That's a carrier. Then this one is a red-green colorblind female. Red-green colorblind female. Write that because it's female because it's XX. So it's red, green, colorblind, female. Then X big and Y, this is going to be a male, a normal male. He's not even a carrier because he doesn't even have that small b, small thing. It's a normal male. Then this is going to be a red, green, colorblind male. This is a red, green, colorblind male. Probability of offspring having red, green color blindness. How many were there? It's 1, 2, 2 out of 4. So it's 2 in 4 or 50%. Do you see how easy it is, students? Do you see how easy the Punnett squares are? My students, are you seeing Antonisha? Everybody, are you seeing? Okay, well done. Cystic fibrosis, blah, 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 represented big F, small f. Actually, don't read too much. 2 heterozygous parents. That's the important thing. What are the genotypes of the parents? They are heterozygous. So it's big F, small f, big F, small f. These are the genotypes of the parents. They are heterozygous. They are gametes, big F, small f, big F, small f. Well done. Then you make the Punnett square. Come on, you can do this. I know that you can do it. Then you put the 3 lines again this way. Then you put big F, small f, big F, small f. Big F meets big F, they cross. Big F meets small f, it's like this. Big F, this is a small f meeting this big F. It will be big F, small f. Small f meets small f, it's small f, small f. What are the offspring genotypes? Big F, big F. Then we have big F, small f. We have again big F, small f. Then we have small f, small f. What are the offspring phenotype? If you have big F and big F, you have no cystic fibrosis. So that is your phenotype. No cystic fibrosis for this one. Even for this one, no cystic fibrosis. The only time you can have cystic fibrosis is if both are recessive. So this is again no cystic fibrosis. Then this person has cystic fibrosis because they have both these. Are we together? Bred together. Predict the phenotypes of the offspring and the phenotypic ratio. So listen, we want the phenotypic ratio. Phenotypic ratio of heterozygous. We know it's 3:1. The phenotypic ratio of heterozygous fruit flies. One has yellow body, which is recessive. Then black body is obviously dominant. So you are going to say 3 black body as to 1 yellow body. That's the phenotypic ratio of these fruit flies. Are we together? Right. Parental phenotypes. In another experiment, this is another breeding experiment. 124:121. That's 1:1, right? Students? Good. We have 120. So here they want the phenotypes of the parents. We talked about it last time. And I was saying to you, for you to get a 1:1 phenotypic ratio, you are going to take one heterozygous, you breed with, they said use big B and small b. You take a heterozygous and you breed with a homozygous recessive. You get a 1:1. Do you remember? So the genotypes will be, they want the phenotypes. This is big B, small b is for what? Represents black body. So they are going to have black body is going to, black body is going to cross with yellow body. The black body will be heterozygous, so it will be big B, small b. They said B to represent the dominant. Then yellow body is small b, small b. The gametes will be big B, small b, and small b, small b. So the danger was if you forget the ratios. You must know the ratios. The ones to remember is that when you see 3s to 1, the parents are heterozygous. When you see 1s to 1, which is here, 124, 121, that's like 1s to 1. That means you have a heterozygous. Are you watching? It's heterozygous and homozygous recessive. Are we together? Antonisha? Everybody? So we already know these parents. How did we know these parents to say heterozygous and homozygous recessive? We saw the ratios in the, you saw that it was 124 and 121. Those are almost the same number. So it's a 1s to 1. So now cross the parents and let's prove it. So you're going to have 1, 2, 3. Then you have 1, 2, 3 like this. And you have small b, and you have big B, small b. You cross big B meets small b, it'll be big B, small b. Big B meets small b, it'll be big B, small b. Small b meets small b, it'll be small b, small b. Small b meets small b, it'll be small b, small b. Can you see it's a 1s to 1? Because there are 2 of this and 2 of this, which will simplify to 1s to 1. So offspring genotypes. What are the genotypes of the offspring? Big B, small b, big B, small b, small b, small b, small b, small b. What are the expected phenotype ratio? 1 black as to 1 yellow. And that is what you have. And you get your marks. So remember this ratio. Are we together, students? Laura? Okay, yes, this is the test course. Ladies, this is the test cross. Yes. Sorry, I hadn't seen. Lag, there's a lag. Yes, this is the test cross. It's like the test cross. When you get a 1s to 1, it's like the test cross where you do a heterozygous with homozygous recessive. That's exactly what you do. You cross with 2 small ones, a heterozygous, then you will be doing a test cross, like a test cross. Anyway, here we have what do they want us to do really? The gametes. So gametes, you just separate this. IA, IO. Here IB, IO, you just put IB, then IO. You just separate. Then you cross. Like honestly, really? 1, cross, guys. 1, 2, 3. Then you put again. Here, 1, 2, 3. Then now you put IA, IO. We can put it even here. IA, IO, IB, IO. IA meets IB, it will be IA, IB. IB meets IO, it will be IB, then IO. B starts before O. IA meets IO, it will be A, then IO. Then IO meets IO, it will be IO, IO. Offspring genotypes, you just take from this bracket here. I mean, they gave you everything, they just wanted you to do this. So now you say the genotypes you take from the boxes. It's IA, IB if it's genotype. Then here we have again IB, IO. Then we have IA, IO. Then we have IO, IO. Then we have the phenotypes. For blood groups, phenotypes will be saying when you have A and B, there is codominant, so it will be AB. When you have this, it's B which is expressed. When you have IO, it is A blood group. IA, IO, A is the dominant one. Then IO, IO, O is the thing. Then the probability that the child will have the blood group AB is 1 in 4, or 25%. Thank you. I think this is the last one. It's not. Oh, okay. There's more. I'm sure now you can try just by looking at this. Here they're saying RH factor is dominant and is represented by capital D. Okay. Then the recessive is by small d. So you know what I look for when I'm reading here? I look for what the parents are, and I look for what letters am I using for the alleles. I don't read anything else. I don't even read all this stuff. So I saw that big D is for dominant, small d is for recessive. And the presence of RH is dominant. So anyway, if a mother is RH negative, that means this big one is not dominant, because if RH, they say RH factor is dominant, the presence of it. So if the mother is negative, that means she has only 2 small d's. If the father is positive, okay, they said an RH negative mom, so she has only small d's, because she mustn't have the dominant one, which is the big D, because it is the presence of it. But the dad is heterozygous, so he is big D and small d. So we come and we put that. The father is big D, small d, lowercase d. The mom is lowercase d and lowercase d. Then you cross them. Capital D for dad meets the lowercase d for mom. It'll be capital D, small d. Then small d here meets another small d. It'll be a small d and a small d. A capital D meets a lowercase d. It'll be a capital D and a small. Then a lowercase d meets this other lowercase d. It'll be lowercase d, lowercase d. Now what do they have? Probability of what? Let's see the probability. They said find the probability of the child being RH positive. If the child is RH positive, that means they have this capital one. And the probability is 2 in 4, or 50%. Is there anyone with a question? Wow, you're doing very well. So this will be it. We're done. Oh, really? May I have my corrections class? Everybody. I wanted to do especially the ones where we were correcting the site for physics. I wanted to do those corrections. I think I'll do them deep in the night, in the AMs. For those who'll be awake, just be checking my YouTube. Should I send the link into the group if I go live, or you check on it tomorrow? What do you want me to do? Do you want to be there in the live, or you just check the solutions tomorrow? Okay, so I'll do that. Thank you, everybody. I'm ending here. We do more corrections. I just want to make sure all assignments are up to date for everybody. So everybody, just do the corrections using the things I did. Make sure your assignments are up to date. I'm going to do the other things, which is the missing one for physics and the missing one for bio, the inheritance one, where it was theory, where I'll be explaining to you protein synthesis, the steps, what you should write and all that. And then the mean for maths. So I'm going to be just going live. You'll be checking. And you see me live when I'm live. Like and subscribe to those who are checking. Goodbye, everybody. Fix the things. Do my corrections if you have not done. Make sure all your assignments are submitted by tomorrow. And we go on to the next concepts. Thank you. Goodbye. All right. Yes, you have a question? If you have a question, you can ask. Pardon, Antonish? I don't know. Your audio is not clear right now. You text. I can't hear you. All right. Bye. Oh, last question. Last question. You can repeat it again. You can just go, where is the problem? Let me see. This one, the presence is dominant. So if somebody is negative, that means the thing is not there. The mom is negative, so that means she doesn't have the dominant one, because the RH factor, the presence one, is the dominant one. So the mom doesn't have it because she doesn't have the, she's negative. So if she is negative, just make sure she doesn't have the dominant one, because the dominant one is for the presence of it. So it's for positivity. So the positive dad is heterozygous though. So he has, yes, he has the dominant one, but he should also have the recessive one because he is heterozygous. Then the mom is negative, which means she mustn't even have the dominant one. So she'll be like this. That's what you do for the mom. And then now you can cross them. I hope you understood that, ladies. I hope you understood that. Did you understand, ladies? All right. Cheers, everybody. Goodbye.